Hello everyone, I am Mohammed Hamama, and this is your ASCP preparation camp. In this camp, we will go through each topic on the ASCP lecture list. Today, we will complete our series on hemoglobinopathies. In today's video, we'll be discussing an important variant known as hemoglobin C. So, let's dive in. Hemoglobin C was the second hemoglobinopathy to be identified, right after hemoglobin S. It is primarily found within the African American population in the United States. In fact, back in 1953, Spate and Rani first reported this disease in its homozygous state, known as hemoglobin CC. The prevalence of hemoglobin C varies among different populations. It is estimated to be around 17% to 28% among individuals of West African descent and approximately 2% to 3% among African Americans. Interestingly, hemoglobin C is the most common non-sickling variant in the United States and the third most common worldwide. Now, let's talk about the structural change that sets hemoglobin C apart. In the beta chain of the hemoglobin molecule, a substitution occurs where glutamic acid is replaced by lysine at position 6. This alteration results in a net charge difference of +2. Although hemoglobin C is inherited in a similar manner to hemoglobin S, its clinical presentation is generally milder. When exposed to low oxygen conditions, hemoglobin C has the ability to polymerize. However, the structure of these polymers differs from those observed in hemoglobin S. In the case of hemoglobin S, the resulting polymers are long and thin. However, with hemoglobin C, the crystals formed within the red blood cells are shorter and thicker. These altered RBC shapes caused by hemoglobin C are less severe compared to hemoglobin S, leading to reduced splenic sequestration and hemolysis. One notable difference between hemoglobin S and hemoglobin C is the occurrence of vaso-occlusive crises. While these painful episodes are commonly seen in individuals with hemoglobin S, they are not typically observed in hemoglobin C patients. Now we'll be exploring the laboratory diagnosis of hemoglobin C. So, let's get started. When it comes to individuals with homozygous hemoglobin C disease, a mild to moderate normochromic and normocytic anemia is commonly observed. There may be slight microcytosis and mild hypochromia present. During peripheral blood analysis, an increase in target cells, a mild to moderate rise in reticulocytes count, and the possibility of nucleated red blood cells can be seen. One characteristic feature of hemoglobin C is the presence of pyramid-shaped or pointed-end crystals within the erythrocytes on the peripheral blood film. These crystals are often found outside the cells and lack a cell membrane. Some cells may contain concentrated hemoglobin within the crystal boundaries. These crystals vary in size and appear deeply stained. They can be visualized on wet preparations by washing and resuspending red blood cells in a sodium citrate solution. The hemoglobin solubility test, commonly used for detecting hemoglobin S, yields a negative result for hemoglobin C. Therefore, definitive diagnosis is typically made using electrophoresis or high-performance liquid chromatography, HPLC. During electrophoresis or HPLC, hemoglobin C presents distinct patterns. In HBCC disease, hemoglobin A is absent, while hemoglobin C is present at levels exceeding 90%. Hemoglobin F is typically less than 7%, and hemoglobin A2 is around 2%. On cellulose acetate electrophoresis at an alkaline pH, hemoglobin C migrates in the same position as hemoglobin A2, hemoglobin E, and hemoglobin O. However, citrate agar electrophoresis at an acid pH separates hemoglobin C from these other hemoglobins. It's important to note that no specific treatment is required for hemoglobin C disease. However, complications may arise if infections occur or if mild chronic hemolysis leads to gallbladder disease. Hemoglobin C-Harlem, also known as hemoglobin C Georgetown, is our next step. What do you think are the unique characteristics of it? So, let's see. Hemoglobin C-Harlem is a hemoglobin variant characterized by a double substitution on the beta chain. At position 6, there is a substitution of valine for glutamic acid, which is the same mutation found in hemoglobin S. Additionally, at position 73, there is a substitution of aspartic acid for asparagine, similar to the mutation found in hemoglobin coralbu. This unique combination of mutations is why it is referred to as Hemoglobin C-Harlem or Hemoglobin C-Georgetown. When performing cellulose acetate electrophoresis at an alkaline pH, Hemoglobin C-Harlem migrates in the same position as hemoglobin C. However, during citrate agar electrophoresis at an acid pH, Hemoglobin C-Harlem migrates in the position typically associated with hemoglobin S. These distinct migration patterns help in the diagnosis of this variant. Individuals who are heterozygous for hemoglobin C-Harlem typically do not experience any symptoms. They appear normal in their clinical presentation. However, patients with compound heterozygosity for hemoglobin S and hemoglobin C-Harlem may have crises similar to those seen in hemoglobin SS disease. To confirm the presence of hemoglobin C-Harlem, a positive result on the hemoglobin solubility test may occur. However, a definitive diagnosis requires hemoglobin electrophoresis or high-performance liquid chromatography, HPLC. During cellulose acetate electrophoresis at pH 8.4, hemoglobin C-Harlem migrates in the position associated with hemoglobin C. On the other hand, citrate agar electrophoresis at pH 6.2 reveals its migration in the position associated with hemoglobin S. Due to the limited number of identified cases, the clinical outcome for individuals homozygous for hemoglobin C-Harlem is uncertain. Further research is needed to fully understand the implications of this abnormality. However, as mentioned earlier, heterozygotes typically appear normal and do not experience any significant clinical symptoms. Hemoglobin E. Hemoglobin E was initially identified in 1954 and is prevalent in about 30% of the population in Southeast Asia. With increased immigration from this region, the prevalence of hemoglobin E has also risen in the United States. However, it occurs less frequently in African Americans and individuals of Caucasian descent. Hemoglobin E is a variant of the beta chain, where glutamic acid is replaced by lysine at position 26. This substitution results in a net charge change of +2, similar to hemoglobin C. However, unlike hemoglobin C, the positioning of the substitution prevents hemoglobin polymerization. However, the amino acid substitution at codon 26 introduces a hidden splice site, leading to abnormal alternative splicing and reduced transcription of functional mRNA for the hemoglobin E-globin chain. As a result, the hemoglobin E mutation presents both a qualitative defect, owing to the amino acid substitution in the globin chain, and a quantitative defect resembling a beta thalassemia phenotype due to decreased globin chain production. Clinical features. In the homozygous state, hemoglobin EE, individuals typically experience a mild form of anemia characterized by microcytes and the presence of target cells. The lifespan of red blood cells is shortened in this condition. However, there are no observable signs of jaundice, hemolysis, or enlargement of the spleen. It is crucial to distinguish homozygous hemoglobin E from iron deficiency, beta thalassemia trait, and hemoglobin E beta thalassemia. Hemoglobin EE disease exhibits similarities to thalassemia trait. Additionally, due to the high prevalence of the hemoglobin E gene in malaria-endemic regions of Thailand, it is believed that the multiplication of Plasmodium falciparum occurs at a slower rate in hemoglobin EE red blood cells compared to hemoglobin AE or hemoglobin AA red blood cells, potentially providing some protection against malaria. Hemoglobin E trait, on its own, does not cause any symptoms. However, when hemoglobin E is combined with beta thalassemia, the disease becomes more severe, resembling beta thalassemia major, and regular blood transfusions become necessary. Laboratory diagnosis. When diagnosing hemoglobin E, it is important to confirm the absence of a positive result on the hemoglobin solubility test and conduct electrophoresis or high-performance liquid chromatography, HPLC. In the homozygous state, hemoglobin E levels are typically over 90%, and there is a very low mean corpuscular volume, MCV, ranging from 55 to 65 femtoliter. The peripheral blood film may show the presence of few to many target cells, and the reticulocytes count is within the normal range. In the heterozygous state, the mean MCV is around 65 femtoliter, slight erythrocytosis may be present, and target cells can be observed. Hemoglobin E constitutes approximately 30% to 40% of the hemoglobin in heterozygotes. During cellulose acetate electrophoresis at an alkaline pH, hemoglobin E migrates together with hemoglobin C, hemoglobin O, and hemoglobin A2. However, on citrate agar electrophoresis at an acid pH, hemoglobin E can be separated from hemoglobin C but co-migrates with hemoglobin A and hemoglobin O. Hemoglobin O-Arab. Hemoglobin O-Arab is a variant of the beta chain where lysine replaces glutamic acid at position 121. This rare disorder is primarily found in Kenya, Palestine, Egypt, and Bulgaria and occurs in approximately 0.4% of African Americans. Individuals who carry this variant typically do not display any clinical symptoms, except for mild splenomegaly in homozygotes. However, when hemoglobin O-Arab is inherited along with hemoglobin S, it can lead to severe clinical conditions similar to those observed in hemoglobin SS. Homozygous individuals with hemoglobin O-Arab experience a mild hemolytic anemia characterized by the presence of numerous target cells in the peripheral blood film. The hemoglobin solubility test yields a negative result in these cases. To confirm the presence of this hemoglobin variant, electrophoresis or HPLC is necessary. On cellulose acetate electrophoresis at an alkaline pH, hemoglobin O-Arab migrates together with hemoglobin A2, hemoglobin C, and hemoglobin E. Therefore, citrate agar electrophoresis at an acid pH is required to differentiate it from hemoglobin C. Notably, hemoglobin O-Arab is the only hemoglobin that shows a slight deviation from the point of application toward the cathode on citrate agar at an acid pH. Now we'll be exploring the unique characteristics of hemoglobin D and hemoglobin G. So, let's dive right in. Hemoglobin D and hemoglobin G are a group of hemoglobin variants that exhibit similar electrophoretic migration patterns to hemoglobin S under alkaline pH conditions. This similarity arises from the fact that their alpha and beta subunits have one less negative charge than hemoglobin A and hemoglobin S at alkaline pH. However, unlike hemoglobin S, hemoglobin D and hemoglobin G do not undergo sickling when exposed to reduced oxygen tension. These variants are often named after the places where they were first discovered. For instance, we have hemoglobin D Punjab and hemoglobin D Los Angeles, both of which are identical hemoglobins where glutamic acid is replaced by glutamine at position 121 in the beta chain. Hemoglobin D Punjab is prevalent in approximately 3% of the population in northwestern India, while hemoglobin D Los Angeles is found in fewer than 2% of African Americans. On the other hand, we have hemoglobin G Philadelphia, an alpha chain variant characterized by the substitution of asparagine with lysine at position 68. HbG Philadelphia is the most common variant among the G hemoglobins observed in African Americans and is also found in Ghana. Both hemoglobin D and hemoglobin G do not exhibit sickling and yield negative results on the hemoglobin solubility test. During alkaline electrophoresis, they migrate in the same way as HbS. However, they can be differentiated from hemoglobin S on citrate agar at pH 6.0. These variants should be considered when encountering a hemoglobin that migrates in the S position on alkaline electrophoresis and produces a negative result on the hemoglobin solubility test. In the homozygous state, hemoglobin D comprises over 95% of the hemoglobin, while normal levels of hemoglobin A2 and hemoglobin F are present. It's important to note that hemoglobin DD can sometimes be mistaken for the compound heterozygous state of hemoglobin D and beta-0 thalassemia. However, differentiating between the two conditions can be achieved by examining factors such as mean corpuscular volume, MCV, hemoglobin A2 levels, and conducting family studies. Heterozygous individuals with hemoglobin D and hemoglobin G variants are typically asymptomatic. In hemoglobin DD disease, which refers to the homozygous state, mild hemolytic anemia and non-progressive splenomegaly may be present. Treatment is generally not necessary for individuals with these variants. In this part, we'll be delving into the intriguing topic of compound heterozygosity with hemoglobin S and another beta-globin gene mutation. So, let's get started. Compound heterozygosity occurs when an individual inherits two different mutant genes located in the same genetic locus, specifically the beta-globin gene locus in this case. In the context of hemoglobin disorders, it means that one parent has passed on hemoglobin S, while the other parent has passed on another abnormal beta-globin gene mutation or thalassemia. The combination of hemoglobin S with other variants like hemoglobin C, hemoglobin D, hemoglobin O, or beta thalassemia can result in varying degrees of hemolytic anemia. Interestingly, when HBS is inherited alongside other hemoglobins like hemoglobin E, hemoglobin G Philadelphia, or hemoglobin Coralbu, these combinations do not cause any clinically significant disorders. Let's now focus on hemoglobin SC disease, which is a prevalent condition characterized by compound heterozygosity. In hemoglobin SC, there are two different amino acid substitutions on each of the two beta-globin chains of the hemoglobin molecule. Specifically, at position 6, one beta-globin chain has a substitution of glutamic acid with valine, hemoglobin S, while the other beta-globin chain has a substitution of glutamic acid with lysine, hemoglobin C. The clinical features of hemoglobin SC disease resemble a milder form of sickle cell disease, SCD. Children with hemoglobin SC may experience delayed growth and development compared to their peers without the condition. However, unlike individuals with hemoglobin SS, sickle cell anemia, those with hemoglobin SC usually do not experience significant symptoms until their teenage years. While hemoglobin SC disease can lead to vaso-occlusive complications seen in sickle cell anemia, the frequency of such episodes is typically lower, resulting in less severe damage. Hemolytic anemia, characterized by the destruction of red blood cells, is moderate in hemoglobin SC disease, and many patients may exhibit moderate enlargement of the spleen, splenomegaly. Notably, proliferative retinopathy, a condition affecting the blood vessels in the retina, is more common and severe in hemoglobin SC disease compared to sickle cell anemia. Additionally, respiratory tract infections caused by Streptococcus pneumoniae are frequently observed in individuals with hemoglobin SC disease. The laboratory diagnosis of hemoglobin SC disease involves several tests. The complete blood count reveals a mild normocytic, normochromic anemia, similar to sickle cell anemia. The hemoglobin level typically ranges from 11 to 13 grams per deciliter, and the reticulocytes count is around 3% to 5%. Upon examination of the peripheral blood film, a few sickle cells, target cells, and intraerythrocytic crystalline structures are observed. These structures, known as SC crystals, protrude from the cell membrane and exhibit characteristics that resemble a combination of hemoglobin S and hemoglobin C crystals. The hemoglobin solubility screening test yields a positive result due to the presence of hemoglobin S. Electrophoretically, hemoglobin C and hemoglobin S migrate in nearly equal amounts, 45%, on cellulose acetate, while hemoglobin F appears normal. On citrate agar at an acid pH, hemoglobin C is confirmed and separated from hemoglobin E and hemoglobin O. In hemoglobin SC disease, hemoglobin A2 migrates with hemoglobin C, but its quantitation does not hold much significance. Determining hemoglobin A2 becomes crucial if there is a suspicion of concurrent hemoglobin C and beta thalassemia in a patient. In terms of prognosis, patients with hemoglobin SC disease tend to have longer life expectancies and experience fewer painful episodes compared to those with hemoglobin SS. It's important to note that hemoglobin SC disease still carries a significant burden of morbidity and mortality, particularly after the age of 30. Hemoglobin S, beta thalassemia, a common form of sickle cell syndrome. So, let's unravel the mysteries of this fascinating condition. Hemoglobin S, beta thalassemia, is a unique condition frequently found in individuals of Mediterranean descent. It stands as the second most prevalent compound heterozygous sickle disorder, right after hemoglobin SC disease. When someone is affected by hemoglobin S beta thalassemia, they often experience clinical symptoms resembling mild to moderate sickle cell anemia. The severity of the syndrome varies depending on the production of beta-globin chains from the affected beta thalassemia gene. Let's dive deeper into the different scenarios. In cases where the beta thalassemia gene fails to produce any beta-globin chains, known as hemoglobin S beta-0 thal, the clinical course resembles that of homozygous sickle cell anemia. On the other hand, if there is production of a normal beta-globin chain, known as hemoglobin S, beta plus, thal, patients tend to have a milder condition compared to those with hemoglobin SC disease. Now, you might be wondering how to differentiate patients with HBS beta thalassemia from those with sickle cell trait. Well, there are several factors that come into play. Firstly, individuals with hemoglobin S beta thalassemia exhibit higher levels of hemoglobin S compared to hemoglobin A. This imbalance in hemoglobin types is a key distinguishing feature. Additionally, increased amounts of hemoglobin A2 and hemoglobin F, which represent different forms of hemoglobin, can be observed in patients with hemoglobin S beta thalassemia. Microcytosis, resulting from the thalassemia trait, is another characteristic feature. This refers to the presence of abnormally small red blood cells. Hemolytic anemia, a condition characterized by the premature destruction of red blood cells, is often present in individuals with hemoglobin S beta thalassemia. When examining the peripheral blood morphology, we may observe abnormal shapes and structures. These peculiarities aid in the diagnosis and differentiation of this particular form of sickle cell syndrome. Lastly, splenomegaly, which refers to an enlarged spleen, is frequently observed in patients with hemoglobin S beta thalassemia. Hemoglobin SD and hemoglobin SG Philadelphia are two intriguing forms of sickle cell syndrome. So, let's get started on this enlightening journey. Hemoglobin SD and hemoglobin SG Philadelphia both belong to the realm of sickle cell syndrome. However, they differ in terms of their genetic composition. Hemoglobin SG Philadelphia is a double heterozygous condition, where individuals inherit hemoglobin S from one parent and hemoglobin G Philadelphia from the other parent. Interestingly, hemoglobin SG Philadelphia is usually asymptomatic due to the presence of hemoglobin G, which allows for sufficient production of hemoglobin A. On the other hand, hemoglobin SD is a compound heterozygous condition, involving the inheritance of hemoglobin S from one parent and hemoglobin D from the other parent. This combination can lead to a wide range of symptoms, from mild to severe hemolytic anemia. Since both beta-globin chains are affected in hemoglobin SD syndrome, some individuals may also experience severe vaso-occlusive complications. In African Americans, hemoglobin D syndrome often arises from the interaction between hemoglobin S and hemoglobin D Los Angeles, hemoglobin D Punjab. To differentiate hemoglobin S from hemoglobin D and hemoglobin G, relying solely on cellulose acetate electrophoresis at an alkaline pH is not sufficient. These hemoglobins comigrate, making it challenging to distinguish between them. Therefore, citrate agar electrophoresis at an acid pH becomes necessary for accurate identification. This method allows us to discern the unique characteristics and migration patterns of each hemoglobin variant. Furthermore, considering the patient's clinical presentation is also invaluable in distinguishing between hemoglobin SD and hemoglobin SG. Understanding the individual's symptoms, medical history, and associated complications can aid in making an accurate diagnosis. When it comes to treatment, managing hemoglobin SD disease is similar to the approaches used for patients with sickle cell disease, SCD. The treatment plan is tailored based on the severity of the individual's clinical condition. Hemoglobin S/O Arab and hemoglobin S/D Punjab. These disorders give rise to severe chronic hemolytic anemia and vaso-occlusive episodes. Let's dive right in. Both hemoglobin S/O Arab and hemoglobin S/D Punjab involve specific mutations at position 121 of the hemoglobin molecule. Normally, glutamic acid resides there, but in these variants, it is substituted. Hemoglobin S/O Arab replaces it with lysine, while hemoglobin S/D Punjab substitutes it with glutamine. Glutamic acid at position 121 plays a critical role in the polymerization process of hemoglobin S. It is located on the outer surface of the hemoglobin tetramer, influencing the interactions between hemoglobin fibers. On cellulose acetate electrophoresis at an alkaline pH, hemoglobin S/O Arab can be mistaken for hemoglobin SC because hemoglobin C and hemoglobin O Arab migrate to the same position. However, differentiation between the two can be easily achieved on citrate agar at an acid pH, providing a clearer picture for accurate diagnosis. Individuals with hemoglobin S/O Arab or hemoglobin S/D Punjab require similar treatment approaches to patients with sickle cell disease, SCD. The management focuses on addressing the complications and symptoms associated with these hemoglobin disorders. Now, let's turn our attention to hemoglobin D Punjab. Similar to hemoglobin S/O Arab, it commigrates with hemoglobin S on alkaline electrophoresis, potentially leading to confusion with SCD. However, neither hemoglobin O Arab nor hemoglobin D Punjab have significant clinical implications, whether in the heterozygous or homozygous form. Let's now explore another intriguing hemoglobin variant, hemoglobin coralbu. This rare type of hemoglobin involves the substitution of asparagine with aspartic acid at position 73 of the beta chain. When hemoglobin coralbu is inherited alongside hemoglobin S, it disrupts the hydrophobic pocket for b-6-valine, affecting the interaction between hemoglobin S fibers. This disruption prevents the polymerization of hemoglobin S, ultimately leading to a lack of symptoms associated with the disorder. Now, it's our time with the intriguing world of concomitant cis mutations with hemoglobin S. Get ready to discover the fascinating stories of three remarkable mutations, Hemoglobin C-Harlem, Hemoglobin S-Antilles, and Hemoglobin S-Oman. Let's begin our journey with Hemoglobin C-Harlem. This unique hemoglobin variant involves two substitutions on the beta chain, the sickle mutation and the coralbu mutation. When individuals are heterozygous for hemoglobin C-Harlem alone, they don't experience any symptoms. However, in the compound heterozygous state with hemoglobin S, the clinical presentation resembles that of hemoglobin SS, commonly known as sickle cell anemia. Hemoglobin C-Harlem produces a positive result on the hemoglobin solubility test and migrates to the hemoglobin C position on cellulose acetate electrophoresis at an alkaline pH. On citrate agar electrophoresis at an acid pH, it migrates to the HBS position, aiding in its identification and differentiation. Now, let's explore two other concomitant cis mutations, hemoglobin S-Antilles and hemoglobin S-Oman. Hemoglobin S-Antilles carries the hemoglobin S mutation, substitution of valine for glutamic acid at position 6, along with an additional substitution of isoleucine for valine at position 23. Similarly, hemoglobin S-Oman also has the hemoglobin S mutation but with an additional substitution of lysine for glutamic acid at position 121. These additional mutations amplify the effects of hemoglobin S, resulting in significant sickling even in individuals who are heterozygous for these variants. Understanding the presence of these mutations helps in accurately diagnosing and managing the associated conditions. Let's now shift our focus to a different type of hemoglobin disorder. Hemoglobin M is caused by various mutations in the alpha, beta, and gamma globin genes, leading to the production of methemoglobin. Methemoglobin, as the name suggests, is a form of hemoglobin where the iron in heme oxidizes, rendering it unable to carry oxygen. This structural abnormality results in methemoglobinemia, characterized by altered oxygen affinity and cyanosis. Hemoglobin M variants involve the substitution of a tyrosine amino acid for the proximal or distal histidine amino acid in the alpha, beta, or gamma chains. These substitutions lead to the presence of elevated levels of methemoglobin, causing the distinctive cyanosis observed in affected individuals. There are several classified variants of hemoglobin M, named after the locations where they were discovered. These include hemoglobin M Boston, hemoglobin M Iwate, hemoglobin Auckland, hemoglobin Chile, hemoglobin M Saskatoon, hemoglobin M Milwaukee 1, hemoglobin M Milwaukee 2, hemoglobin FM Osaka, and hemoglobin FM Fort Ripley. Diagnosis of hemoglobin M is typically done through spectral absorption analysis of the hemolysate or hemoglobin electrophoresis. The absorption spectrum peaks at different wavelengths, aiding in the identification of the specific hemoglobin M variant. Electrophoresis on agar gel at pH 7.1 allows for clear separation and further confirmation can be obtained through techniques like HPLC or DNA-based globin gene analysis. Treatment for hemoglobin M is generally not necessary, but accurate diagnosis is crucial to avoid inappropriate treatment for other conditions, such as cyanotic heart disease. Have you ever heard about unstable hemoglobin variants? Can you take a guess? Leave a comment and let us know. Don't forget to subscribe, activate notifications, and share this video with your friends. Unstable hemoglobin variants occur when our globin genes undergo genetic mutations, leading to the production of hemoglobin molecules that tend to precipitate within our bodies. This intriguing phenomenon gives rise to unique structures called Heinz bodies and results in a specific type of anemia known as hemolytic anemia. With over 140 known variants of unstable hemoglobin, it's a complex world out there. The majority of these variants affect the beta chain, followed by the alpha chain, while only a few variants have an impact on the gamma and delta chains. Each variant carries its own distinctive characteristics. Now, here's a fascinating fact: although most unstable hemoglobin variants do not have significant clinical implications, many of them exhibit an increased affinity for oxygen. It's like a secret superpower hidden within our very own red blood cells. However, approximately 25% of unstable hemoglobins are associated with hemolytic anemia, which can range from mild compensated anemia to severe hemolytic episodes. It's a delicate balance between our genetic makeup and the well-being of our blood. In the past, this form of anemia was known by different names, such as congenital nonspherocytic hemolytic anemia or congenital Heinz body anemia. But today, we more accurately refer to it as unstable hemoglobin disease. Unstable hemoglobin disease typically reveals itself at birth or shortly after, depending on the specific globin chains involved. It's important to note that this condition is inherited in an autosomal dominant pattern, meaning that affected individuals are heterozygous for the mutation. Homozygosity for these mutations, on the other hand, is generally incompatible with life. The instability of the hemoglobin molecule can be caused by a variety of factors. It's like a delicate symphony, where even the tiniest change can have significant consequences. Factors such as the substitution of charged amino acids within the molecule's interior, the switch from polar to nonpolar amino acids in the hydrophobic heme pocket, or the alteration of contact points between different subunits of the hemoglobin molecule, all play a role in shaping these unstable variants. Additionally, specific amino acid replacements in the alpha helix section of the alpha chain or the deletion/elongation of certain parts of the primary structure of hemoglobin can also contribute to its instability. It's truly remarkable how our genetic code can lead to such intricate variations. Clinical features. Unstable hemoglobin disorders typically reveal themselves in early childhood. It's during this time that patients present with hemolytic anemia, jaundice, and an enlarged spleen, also known as splenomegaly. These symptoms serve as a signal, alerting us to the presence of something extraordinary happening within our bodies. The severity of the anemia experienced by individuals depends on just how unstable their hemoglobin molecules are. Here's an intriguing twist: unlike normal hemoglobins, unstable hemoglobin tends to precipitate both inside and outside of our red blood cells. It's like a dance between our genetic makeup and external factors like drug intake, heat, or cold. This precipitation gives rise to unique structures called Heinz bodies, fascinating formations within the RBCs. The hemoglobin attached to these bodies clusters together, forming a bond with band 3 and even attaching to autologous immunoglobulins. These Heinz bodies don't just sit idly by. They have an impact on our immune system. By activating macrophages, they alert our body's defense mechanisms to their presence, triggering a response that's designed to maintain our well-being. However, the journey of Heinz bodies doesn't end there. They can become mechanically trapped in the spleen, our remarkable organ, leading to a shortened lifespan for our RBCs. Moreover, the oxygen affinity of these cells is also affected. It's like a subtle shift in the balance of our respiratory system, altering the way our blood carries and delivers oxygen throughout our body. Now, let's take a moment to explore some of the specific unstable hemoglobin variants that exist within this intricate landscape. The most common form of unstable hemoglobin is hemoglobin colon, a name that echoes through the corridors of our genetic heritage. But it's not alone. There are other variants, such as hemoglobin Hammersmith, hemoglobin Zurich, hemoglobin Gun-Hill, and hemoglobin Hammersmith, each with its own unique characteristics. What's truly remarkable is the significant variability in the degree of instability among these variants. Take hemoglobin Zurich, for example. This variant requires the presence of an oxidant for significant hemolysis to occur. When it comes to diagnosing unstable hemoglobin disorders, the journey begins in the laboratory. It's here that scientists and medical professionals employ various tests and observations to uncover the secrets hidden within our blood. One important aspect of laboratory diagnosis is examining the morphology of red blood cells, RBCs. Interestingly, the appearance of RBCs can vary. Some may appear normal, while others exhibit slight hypochromia, which is a reduction in color, and prominent basophilic stippling, the clumping of ribosomes within the cells. Before undergoing a splenectomy, a surgical procedure to remove the spleen, patients typically have hemoglobin levels ranging from 7 to 12 grams per deciliter. Alongside this, their reticulocytes count, a measure of young red blood cells, may range from 4% to 20%. Even after splenectomy, although anemia is corrected, an elevated reticulocytes count, known as reticulocytosis, persists. One intriguing observation in the laboratory involves the visualization of Heinz bodies. These are abnormal structures formed by precipitated hemoglobin. They can be seen using a supravital stain, which highlights their presence. After splenectomy, Heinz bodies become larger and more numerous, adding another piece to the puzzle. Another challenge in diagnosing unstable hemoglobin variants arises from their similar migration patterns to normal hemoglobin during electrophoresis. To overcome this, additional tests are employed to specifically identify these variants. One such test is the isopropanol precipitation test. This test takes advantage of the fact that an isopropanol solution at 37 degrees Celsius weakens the bonding forces of unstable hemoglobin molecules. If unstable hemoglobins are present, rapid precipitation occurs within 5 minutes, followed by heavy flocculation after 20 minutes. In contrast, normal hemoglobin begins to precipitate only after approximately 40 minutes. Another technique used is the heat denaturation test. Heat-sensitive unstable hemoglobins exhibit a flocculent precipitation when incubated at 50 degrees Celsius for 1 hour, while normal hemoglobin shows minimal or no precipitation. The process of splenectomy itself can provide valuable insights. It can result in the appearance of a significant number of Heinz bodies, amplifying the clues left behind by these unstable variants. Even in individuals with intact spleens, a longer incubation period and the addition of an oxidative substance, such as acetylphenylhydrazine, can induce the formation of more Heinz bodies in unstable hemoglobins compared to individuals with normal hemoglobins. Other sophisticated methods come into play as well. Techniques like isoelectric focusing can distinguish between hemoglobin variants with slight variations in their isoelectric points, unlocking further details of their unique identities. And the analysis of globin chains can be performed using advanced techniques like high-performance liquid chromatography, HPLC, or DNA-based globin gene analysis. Now we embark on a fascinating exploration of hemoglobins with abnormal oxygen affinity. Within our bloodstream, there exists a multitude of hemoglobin variants, each with its own unique characteristics. Among them, the majority display increased oxygen affinity, a phenomenon often associated with familial erythrocytosis, a condition marked by an excessive number of red blood cells. Conversely, some hemoglobin variants exhibit decreased oxygen affinity, which can lead to mild to moderate anemia. To understand these deviations, let's take a closer look at normal hemoglobin, also known as hemoglobin A. As this remarkable molecule transitions from a fully deoxygenated state to a fully oxygenated state, it undergoes conformational changes that affect its interaction with oxygen. These changes influence hemoglobin's affinity for oxygen and its overall function. In the deoxygenated state, often referred to as the 10th state, normal hemoglobin displays a low affinity for oxygen and other heme ligands. However, it has a high affinity for allosteric effectors like Bohr protons and 2,3-bisphosphoglycerate, which play crucial roles in regulating its function. On the other hand, in the oxygenated state, known as the relaxed state, hemoglobin exhibits a high affinity for oxygen while having a low affinity for Bohr protons and 2,3-bisphosphoglycerate. This transition from the 10th state to the relaxed state involves significant structural alterations that greatly impact hemoglobin's function. Now, imagine if an amino acid substitution occurs, compromising the stability of the 10th structure. In such cases, the transition to the relaxed state occurs earlier during oxygen binding. This results in increased oxygen affinity and a decrease in heme-heme interaction or cooperativity within the hemoglobin molecule. One fascinating example of a variant in the B chain is hemoglobin C, an unstable hemoglobin variant. Hemoglobin C carries amino acid substitutions at critical sites that play a crucial role in hemoglobin function. These alterations disrupt the delicate balance of oxygen affinity, adding yet another twist to the intricate tapestry of hemoglobin variants. Hemoglobin C is just one among the vast array of hemoglobin variants that challenge our understanding of oxygen affinity. From hemoglobin C to countless others, each variant carries its own unique amino acid substitutions and structural modifications, altering their interaction with oxygen and reshaping the complex dynamics of our bloodstream. Hemoglobins with increased oxygen affinity. Hemoglobins with increased oxygen affinity follow a pattern of inheritance known as autosomal dominant. Individuals affected by these variants typically possess an equal amount of normal hemoglobin, hemoglobin A, and the abnormal variant. However, there are exceptional cases where the proportion of abnormal hemoglobin surpasses 85%, especially in compound heterozygotes for certain variants and B thalassemia. Over 90 high-affinity hemoglobin variants have been identified so far. These variants exhibit a reduced efficiency in releasing oxygen when needed, which can lead to a condition known as hypoxia. In response to hypoxia, the kidneys sense the oxygen deficiency and produce higher levels of erythropoietin, a hormone that stimulates the production of red blood cells. This compensatory mechanism results in a condition called erythrocytosis, characterized by an increase in the red blood cell count. Unlike unstable hemoglobins, which may also display abnormal oxygen affinity, high-affinity variants do not cause precipitation and hemolysis within the body. Additionally, there are no noticeable abnormalities in the morphology of red blood cells. In most cases, individuals with high oxygen affinity hemoglobins do not experience physical symptoms apart from a ruddy complexion. Asymptomatic, they often discover their condition during routine examinations, where elevated levels of red blood cell count, hemoglobin, and hematocrit are observed. White blood cell count, platelet count, and peripheral blood film findings usually fall within the normal range. To establish a diagnosis, hemoglobin electrophoresis can be employed. This method may reveal an abnormal band separate from the normal A band on cellulose acetate for some variants. However, the absence of such a band does not rule out increased oxygen affinity. In such cases, alternative techniques like citrate agar, pH 6.0, or gel electrophoresis may be necessary to separate the abnormal hemoglobin. Measurement of oxygen affinity is crucial for a definitive diagnosis. It's essential to note that patients with high oxygen affinity hemoglobins can lead normal lives without requiring specific treatment. However, accurate diagnosis is crucial to avoid unnecessary treatment for conditions such as myeloproliferative neoplasms or secondary erythrocytosis. Now, let's shift our focus to hemoglobins with decreased oxygen affinity, an intriguing contrast to their high-affinity counterparts. These unique variants possess a fascinating characteristic of rapidly releasing oxygen to the body tissues, resulting in a normal or slightly decreased concentration of hemoglobin. In some cases, this decreased oxygen affinity may contribute to mild anemia. One notable example of a hemoglobin variant with decreased oxygen affinity is HB Kansas. This variant is caused by an amino acid substitution where asparagine is replaced with threonine at position 102 of the B chain. Such substitutions contribute to the altered oxygen affinity observed in these variants. To identify hemoglobin variants with decreased oxygen affinity, techniques like starch gel electrophoresis, high-performance liquid chromatography, HPLC, or DNA-based analysis of the globin gene are utilized. These methods aid in the detection and characterization of specific abnormal hemoglobin variants with decreased oxygen affinity, shedding light on their distinctive properties. Stay up to date with our latest videos by hitting that subscribe button and activating notifications. Be the first to know when we release new and exciting content. Don't keep all this valuable information to yourself, share this video with your friends who might find it interesting and beneficial. If you have any questions or comments, feel free to leave them down below. Until next time, take care and goodbye.